Welcome everyone to the first Q&A interview of the CRS Gene Delivery and Gene Editing Focus Group. My name is Witzigmann and my today's guest is the legendary Pieter Cullis. Welcome, Pieter.
It's good to be here.
Yeah, it's great to have you here. When we look to the career and your journey through the nanomedicine, like you have made fundamental achievements, just mentioned some. So Pieter has more than 300 research articles, more than 50,000 citations. He has founded several companies, among these Acuitas Therapeutics, Integrated Nanotherapeutics, Precision Nanosystems.
Inex Pharmaceuticals.
Yeah, we can chat about this. And yeah, you also co-developed many drugs and most importantly, co-developed on Petro, the first RNAi therapeutic.
Right.
So, how did everything start? How did you go into science?
This goes obviously way back, 50 years ago. So I took physics as an undergraduate, and which I quite enjoyed. And then, like anybody else who graduates with a bachelor's degree, didn't know quite what I was going to do with my life, and it seemed to be an obvious thing to go on to graduate school. So I went on to graduate school in physics, and there I found that the physics is a very challenging discipline, and there were people there that were a lot smarter than me. It's also a very not as social a discipline perhaps as others. And so by the time I got to the end of my PhD, I was really looking quite hard at going into another branch of science. And it so happened that one of the lead professors in the department had organized a course on membranes. He didn't know much about membranes either, particularly biology. He was a physicist. But he would bring in various people from the biological departments to talk about membranes. And I got the impression there were a lot of unsolved problems there. So I switched fields. I then went on to do a postdoc in Oxford, working for a guy named Rex Richards, who actually introduced NMR into England. He worked at Harvard for a while, and then went on to go back to introduce NMR to the British. This was in the 50s. Anyway, so he had just retired for being head of the Department of Chemistry at Oxford, and they had this tradition. Well, he hadn't retired; he'd gone on to take another position in the university. They had the tradition in the chemistry department that they had to move out of the department if they wanted to continue running a lab. So he moved his lab to the biochemistry department, which turned out to be very fortuitous. But he brought all his NMR machines with him. And so that meant that when I got there, I knew a bit about NMR from my PhD studies. Magnetic resonance is what I was studying, and so I was in a good position to start applying magnetic resonance (NMR) to biological systems. And the interest of Rex Richards, and particularly a guy named George Rada, who at that point was a professor in the biochemistry department, was in the biological membranes. And so we started to use NMR, phosphorus NMR, really for the first time anybody had used it, to look at the properties of obviously lipids in membranes. Because they're phospholipids, they have a phosphorus nucleus. And so it was a good way of getting into a field that I initially knew very little about.
And how did you then make the step to nanomedicine?
Well, this was actually fairly, again, one tries to simplify one's life. Anybody who's studied biological membranes realizes that they're really complicated, with not just hundreds of proteins, but thousands of different lipids present in those membranes. And so the only way that you can make sense of what the physical properties might be of those various lipid components is to extract them and to make model membranes out of them. And so the model membranes, of course, are things like sonicated vesicles, unilamellar vesicle systems, which you can then study in detail to see what the properties of the particular lipids are, the structural properties and the permeability properties, fusion properties, etc. And so that's what I did, looking at lipid polymorphism, the ability of lipids to adopt different structures using NMR initially, or using NMR almost completely actually at that time. And then I gradually got more and more interested in the model membranes themselves. This was in the late 70s, early 80s, and so the idea of using lipid-based systems as delivery vehicles was just coming into consciousness. And in other words, maybe we could use these things to deliver drugs to where we wanted to deliver them. And I found myself getting increasingly interested in that application as opposed to the basic physical properties of lipids in membranes, although I did continue that for some time, that area of research. But anyway, that was the trajectory, is trying to understand lipids and the roles, the physical properties, functional roles of lipids in membranes, and then using model membrane systems to get to those, to get some insight into that, and then finding that, hey, we can use these things in very direct applications, which I found very interesting. It's a very interesting way to go.
Yeah. And how did you know, like, what kind of questions you should ask? Because it's really basic research, comes from, you're a trained physicist, and then some of the things I have to check, like the shape hypothesis, which is now, like, important for gene delivery and, like, the endosomal escape of lipid nanoparticles. How did you know at that time, like, what question to ask?
Well, as I said, we were not thinking of these systems in terms of being delivery systems. We were thinking of them in terms of well, how can we understand why these lipids are in membranes and what they're doing? So that was the questions we were asking. We were asking, for example, the phosphatidylethanolamine found in biological membranes will put it by itself and will adopt this non-bilayer structure, this hexagonal H2 phase structure. So what is it doing in a membrane? And so we were, that was the fundamental driver. It turned out, of course, that that understanding was quite useful in terms of designing systems that can fuse more readily, that can break out of an endosome.
Yeah.
But so we didn't have any feeling that was going to be the case.
Yeah.
We were looking at it in those early days.
And what do you think have been the barriers or challenges you faced during that time, like going into gene therapy? And
well, one of the things that is a bit of a handicap, or has been a handicap, in the call it nanomedicine area or the drug delivery area, using various nanoparticles, has been people often use systems that are not very well characterized, and this was something that. We as a group had always tried to do, is to work always with systems that are there where we really understand what the system is before we ever go near an animal. And we know we can make it the same way each time, and that we can control various variables, like we have very efficient drug loading processes. The manufacturer is scalable. In other words, we'll be able to make large amounts if necessary. That means that you eliminate at least one set of variables from the experiment. If you're doing a biological experiment, particularly when you're injecting something into an animal, the number of complications are huge, and you really want to remove the complication that maybe your system is changing from one point to another. So working with very reproducible, very well-defined systems was something we started off with right from the beginning. I mean, that was an essential feature.
Yeah, and when we think about ONPATTRO, what do you think there have been the crucial discoveries to make this happen?
Well, with ONPATTRO, there was quite a number of interesting and really, you can only say it's really good luck that we managed to get to them. But the first one was that we knew from all the work we'd done with the small molecule systems, where we were using lipid nanoparticles, liposomal systems, as drug delivery agents for cancer drugs. But we knew in order to get any reasonable circulation lifetime and tissue distribution, we had to have relatively neutral carriers. And of course, this is a big problem when we're trying to extend that to nucleic acid based systems. particularly if we're using cationic lipids in order to associate them with the nanoparticle. And so before we started off doing anything, we said, okay, well, how can we beat this problem of of having a relatively neutral system, but also using being able to encapsulate the RNA or DNA inside a nanoparticle. And so that's where we hit on the idea of using an ionizable cationic lipid, which we had developed for a completely different application. We could flip an ionizable cationic lipid across a membrane in response to a pH gradient. And so we were interested in lipid asymmetry and some of the consequences. So we had an ionizable cationic lipid sitting there in the refrigerator. So we thought, okay, well, maybe we can encapsulate the nucleic acid in a lipid nanoparticle at low pH, where the ionizable cationic lipid is charged. And then maybe if we bring that back up to neutrality, we'll maintain encapsulation. But of course, we'll have a nanoparticle that had much less of a charge on the surface, much less toxic, because cationic lipids are toxic as well. And that turned out to be the case. But as I say, we got lucky in the sense that it then turned out that the detailed design of the cationic lipid, and particularly the acid dissociation constant, the pKa, that we could really tune that and find that the potency of these systems could be dramatically influenced by what that pKa was. But I should say that the first step was to get systems that were well defined, and we could get good loading into nanoparticles and have a relatively neutral exterior, and it was a manufacturable. We could synthesize these systems with some ease, and we did all that before we then went on to the next step. But one of the things we had learned from our work with lipid nanoparticles and small molecule drugs is that any results we got in vitro were they didn't really help very much. There was no correlation between potency in the in vitro experiments and extending that to in vivo. So as soon as we got a system where we had the requisite encapsulation size, neutrality, etc., we moved into animal models, and this was really a very good thing that we did that, because we wouldn't have found out a lot of other, the advances would not have been made if we'd been working in vitro trying to optimize these systems further.
And how crucial was that you founded several companies, starting really early with Lipec?
Lipec's biomembranes.
Yeah,
yes.
And then through the discovery of and development of on Petro. How crucial was during all these steps?
It's absolutely crucial, and one of the only reason that we've been reasonably successful in the drug delivery field is that we've managed to maintain a nucleus of people that work together for a very long time, and locally here in Vancouver. And so when I started off my lab here at UBC, University of British Columbia, in 1978 or 78, I think. Within a couple of years, I'd got the 3 or 4 postdocs who were really, we were kind of very keen and very motivated to push forward. So this was Mick Hope, Tom Madden, Marcel Bally, and Lawrence Mayer. Who were either graduate students or postdocs, mainly postdocs in the lab. And one of my real driving things has been that, it annoyed me to graduate students and then have them go back east to Eastern Canada or to the States or wherever to get a job. That we really needed to have something going locally. And that was the era when biotechnology companies were being started off, like Genentech and others, and Biogen. And so I said, okay, well, why can't we start companies ourselves, and then keep people locally and start to grow things locally, which is what we did, and that's really been a large reason for the success we've had. It's because we've maintained a very high-level expertise locally. And it's difficult to do that in a university setting. It's difficult to do it in a company setting too.
Yeah.
But we succeeded in doing that, and that meant that we've really managed to keep a lot of expertise local, and have managed to grow an industry, a local industry that now is over 300 people that are employed locally in the lipid nanoparticle field as a result of our efforts. Anyway, so that's starting companies has been essential, because it's also meant that we never raised a lot of money for taking drugs through clinical trials, etc. You can't raise that kind of money in a university setting. It's also meant that we can have affiliations, company-to-company affiliations with other companies, such as Alnylam, that's the company in Boston that is the siRNA, RNAi leader as a therapeutic. And so those sort of associations from a company-to-company point of view with, say, INEX or Acuitas are easier to set up than ones from a university setting. So anyway, it really is, for those people that are thinking about getting the results in their laboratory and translating those into the clinic, you really have to think hard about starting a company, because it facilitates that process enormously.
Yeah.
And it's not an easy thing to do, but it's extraordinarily difficult to do it to get the stuff going on in your lab into the clinic without doing that.
Can you mention staying with the companies, saying 2-3 highlights during these?
Oh yeah, I mean for those people that, as I said, it's a good idea to start companies, but once you get into a company environment, it's pretty rough. Within a university setting or an academic setting, there's lots of, we know the rules. We have to publish, and we have to get grants, and then we'll get ahead. It's a bit different in the world of the industry, because there really aren't very many rules, and it's a very dog-eat-dog. It's quite a vicious environment, and so you have to be prepared for companies starting to fight with one another, lawsuits and stuff like that. When you're there's HR issue. We had a company of well over 100 or 150 people. It's a bit more than that at one point, and then you get into human resource issues and all of that kind of thing. And you have to be. It's very difficult to choose the right CEO. Sometimes you do, and sometimes you don't. These are very important decisions that will really affect the way things go. So for those of us that have an academic background, there's actually very little training or experience that you have when you move into that realm. Luckily, people that are in business don't have much of a clue either. So it's I'm being facetious there, but it's kind of in a way true that managing a enterprise, a company in a company environment can be done in many ways, and so there's no one set path through the process. All I know is you got to start something off, and you go in one direction. Maybe that doesn't work, and but in a company environment, at least you can change course, and then over a period of a few years, you'll then get to the point where something you'll get things settled down, and the company is actually doing what it's doing something that is is of value. But it's a very challenging environment. But the really fun thing is the team aspect that it builds. In an academic setting, of course, you're more you do research as an individual. In a company, you're part of a team, and that is a lot of fun.
Yeah.
Particularly at the early stages.
Yeah.
It's terrific fun because it's you against the world, and you have to do everything. Everything from presenting to potential investors, to the things like HR, setting up the structure of the company, writing business plans, you name it. It's and that's, you're learning a lot all the time, very useful things all the time.
Let's chat a bit about like all these companies you founded, all the teams you created. They ended up in like several approved drugs. And let's chat about Onpattro. It was approved 2018, was a breakthrough, the first RNAi therapeutic. What do you think was crucial for this development of Onpattro, and how did this change the field of gene therapy?
As you're probably aware, I mean, the field of gene therapy has a rather checkered history. Not checkered, but it's a difficult area. I think there's been 2,500 clinical trials since about 1990, and there's only about 4 approved drugs, FDA-approved drugs that have come out of all of that. And there's been a number of notable failures. And so the real issue has been the use of viral vectors. There is a problem, of course, is one of limited payload. You can only put about 5 kilobases in, and then you've got a problem. The ability to only inject once, because the next time around you've got an immune response. Manufacturing viruses, it's just got all sorts of issues. It goes on, and you end up with very expensive products at the end. Now, I think that the non-viral approach, getting that to work is completely revolutionary because you're using materials that are very inexpensive. You have a manufacturing process that is well defined and well laid out, and you can have any size. You can have 10 kilobases if you want. And also, of course, you eliminate the problem of the immune response. The list of advantages goes on and on. So the breakthrough of Onpattro, significance really is that the same technology appears to be able to be applied to much bigger entities, messenger RNA, etc., which really enables the whole, as far as I can see, the whole of gene therapy, including gene editing. And so we can now express any protein we want, we can silence any protein we want, and we can start with a gene editing approach to edit any protein we want. Now, at the moment, we're confined to the liver as a target tissue where these things work the best. But already we're showing that for, say, intradermal, sub-Q, IM, for vaccine applications, that these are working very well. I suspect that within the next few years we'll start to get systems that are able to have access to other tissues, immune cells, bone marrow, the list goes on and on. So the ability to perform gene therapy in very many forms is going to be extended to all those tissues. Now, one of the points that's just amazing to me is how quickly you can devise a medicine using this approach. I mean, once you know, for example, that a child has had a hereditary disease where they're not making a particular protein, then you can say, okay, well, let's get that made in the liver. You can probably manufacture the messenger RNA in a time on the order of weeks, say a couple of months, absolute maximum. You can package it into a lipid nanoparticle in a day, and you have the medicine ready right then for that individual. This just contrasts so enormously with the very convoluted and long-term and expensive process that we have for small molecule drugs, and also to a large extent with viral vectors, where you have to do a lot of engineering in order to be able to get something that may or may not work very well. So I think it really does change the game a tremendous amount.
And what do you think, when we look at the current situation and the future and developing the full potential of lipid nanoparticles, what could be the barriers or challenges we face?
Yeah, the challenge is always one of potency and toxicity. What we did with Onpattro, we worked in a mouse model, a factor 7 mouse model where we were knocking down factor 7 as a surrogate in hepatocytes, and increasing the potency largely by optimizing the ionizable cationic lipid. And so we ended up with a therapeutic index of over 1,000. In other words, we had to give 1,000 times higher dose before we saw some really, potential toxicity in the mouse model. And that's it's somewhat less in non-human primates and people, but you need that kind of big therapeutic window. So that's always going to be the barrier, or not going to be. You have to. The same with any other therapy, right? I mean, if you have a small molecule drug, then you give too much of it, you're going to have problems. But I have a feeling that we're going to be able to extend that window, say 1,000 therapeutic index for the gene silent for RNAi, siRNA. I have a feeling that we're going to be able. That's where the refinement's coming in, and we'll get better and better at it. And really get to the point where these toxicities, which are one of the nice things about the technology, is you're going to get the same toxicities, at least from the carrier, irrespective of what the cargo is. And so, from a clinician point of view, it's a much easier thing to handle. And of course, clinicians are now very much attuned because of the. There's now 10 lipid nanoparticle drugs that have been approved for clinical use, and so most of the toxicities that arise are well characterized and can be controlled well in terms of clinical utility. But as I say, I think the potency of these systems, we're not at the end of the road yet by any means. That would be able to improve it further without increasing the toxicity. Or even reducing the toxicity. One can see ways of doing that. So it's, it's pretty neat. It's a very exciting time. As I say, it really is revolutionary, being able to design a medicine and make it in a time on the matter of weeks. This kind of thing is going to turn the regulatory. I mean, obviously it has regulatory issues, and it's going to be from all sorts of points of view, it's going to change the game a lot. But I think it's going to change it in a very positive way.
And if we think about long-term toxicities, like when we think about genome editing, do you think we have sufficient knowledge yet?
Oh no, the gene editing approaches are going to have to get very, very precise. It's what in which they aren't, to my view, quite precise enough yet. We can't be as predictive. Of course, if you're suffering from a nasty hereditary disorder, I mean, maybe you're going to take your chances. But for a lot of things, we're not quite as precise as we might like to be. But for siRNA, where you're silencing a gene, or for messenger RNA, where you're expressing a gene, and you're not really accessing the nucleus, and you're not altering the host genome, the long-term toxicities, I think, are much easier to handle. And of course, you'll be able to, if you're having a deleterious effects, well, you stop giving the drug. So you can manage issues in a more straightforward way.
Yeah, and let's say we want to fully develop the potential of lipid nanoparticle technology for gene therapy. What is, in your opinion, the call to action for scientists, clinicians, government?
Yeah, there's a number of things. I think the first thing is to realize this is going on. It's not generally realized now that we're starting to move into this era where gene therapy is going to be a common kind of therapeutic. It's always been kind of on the horizon. Like pretty soon we're going to get gene therapies. Well, as I said, there's 4 gene therapies that have been approved by the FDA. Right now we're only good at, as I said, the liver. Which there's 200 diseases associated with the liver, so this gives you a lot of running room. I think the real changes the real awareness is going to come in as we're able to access more and more tissues. That's going to be bone marrow and leukemia and all sorts of applications there. Once we access the immune system, various immune disorders, and all of these things, you can manipulate the immune system. Obviously, you can manipulate pretty much anything in terms of just fighting disease. So as we get one tissue after another, That's going to be a big deal. The other big deal is going to be for rare diseases. And this is particularly rare genetic diseases, which I think accounts for a good proportion of childhood illnesses. And so once you identify what protein is not being made by this individual, you're going to be able to devise a medicine very quickly. Now, this is going to cause, from a society point of view, I think, an interesting reaction because people that are desperate when something is affecting their child, and that child may die as a result of some genetic problem. And when you have the ability, as I say, to put together a medicine that may be therapeutic for that child, and have good reason to think that, the pressures on the medical system to allow that to happen are going to be enormous. And so I think that's going to be something that really does also change the rapidity with which gene therapy gets mainstream.
And do you think we should, as scientists, work closely together with government, regulatory agencies, industry?
Well, I think it's becoming more and more apparent that we have to, particularly as I work obviously in a biochemistry department, and you're used to, thinking, okay, anything I discover, that'll have a ramification therapeutically maybe 10 years from now or whatever. But now, of course, anything you discover that might have something to do with regulation of a gene or the function of a gene can be, conceptually at least, made into a therapeutic almost immediately. And so the need to start to integrate that with the medical system and the Health Canada and the FDA is becoming more and more apparent. I mean, certainly the FDA is really getting on board with this. They're realizing gene therapy is coming down the track pretty quickly, and have set up they're quite helpful in terms of guiding you through, say, a clinical trial process, or the process by which these drugs can get accepted. But from the scientist's point of view, you need to become more and more aware of those demands because what you discover today may be a drug tomorrow. So.
Yeah. And with regard to young scientists, PhDs, postdoc, what would you suggest them? Like, how can we make an impact?
Yeah, well, I do think this is early days, obviously, in this whole field. I think for all of us, you start off your research, but you always have this feeling like, I really want to see this being of use, someday, as a to help treat whatever disease one might think of. And I say for young scientists right now, you now have the opportunity in a very direct way to be able to do that and have some expectation that within some reasonable time period, you'll be able to get your idea into a clinical setting. So it's enormously exciting for people in the life sciences. The opportunity really is there. That it really wasn't there, hasn't been there up till now.
It's definitely motivating.
Well, I think it's highly motivating. It's not often. It's a gold rush time. Because so many diseases now, you can say, almost as soon as the genetic disease, the hereditary disease, as soon as you know what the problem is, you can design a medicine. But similarly, for almost any other disease, as soon as you say, okay, well, what's a pathway, biochemical pathway can we affect that might be useful to treat that disease? There's a lot of validated targets. Well, you can devise something to either express a particular protein or silence one very quickly. And so that's where the application, everything from cancer to the immune diseases, you name it, infectious disease, just keeps on coming.
Yeah.
So I think the coronavirus is going to be solved by an mRNA that codes for the coronavirus. And put that in a lipid nanoparticle. I think Moderna has already been contracted by the U.S. FDA to start making that. So and you can turn this around in a matter of a month. So it's really fast. A really fast process. So it's a very exciting time. So I would advise anybody in their early career stage in the life sciences to really consider this area, because it's going to be the medicine of the future as far as I can see.
Yeah. We went now quickly through like 40 years of monumental discoveries. Is there anything like I haven't asked what you think should be touched? Any topic?
Let's see. I guess I did mention it, but the need for whatever you're putting into an animal model or into with the intent of, but the need for very rigorous characterizations of these systems, so that you know that you can make the same damn thing, in the same way forever, and you can make lots of it. It's held the field back a bit, but that's probably our singular advantage, is really having well-characterized systems that are 100% trapping efficiency and all the rest of it. The other one is the use of animal models. I mean, people don't necessarily, you don't necessarily like to use an animal model for obvious reasons. But it's very difficult to predict what's going to happen in vivo from an in vitro experiment. And the big example to me there is, for Onpattro, this relies on association with apolipoprotein E to trigger uptake into hepatocytes. The chylomicron remnants use that pathway, and so we've hijacked that pathway. Now, we would never have discovered that. But from in vitro experiments. So these natural targeting, if you want to think of it that way, events only can come by working with an animal model. And it's surprising, actually, how well that carries through from mouse to human primate to people. The same mechanism enables to be very predictive. So that's those are a couple of things that stand out.
Maybe as a last point, you are the scientific director and CEO of the Nanomedicine Innovation Network. It's a big network of clinicians, scientists from different fields. How important do you think this will be for the future potential of lipid nanoproduct development?
Well, one of the things that you note for this approach is it's multidisciplinary. And so we need people that work on a particular disease and have the animal models associated with that. We also need the physicists and engineers and chemists who can help us with various aspects. And so these kind of interdisciplinary networks are essential. If you're just working with the people that have the same background as you, it's much less fertile. For example, one of the things that we're going to be aiming for, who knows how far out it might be, but it's all very well to get gene expression or be able to practice gene therapy, we'll say, in the bone marrow. But it's very likely we're also going to be hitting the liver. I mean, and it's very hard to get to the bone marrow without getting some to the liver. And so as a longer term aim, we're going to want to have site-specific gene therapy, and that's only going to happen. I mean, so you're going to have to essentially have systems that aren't really potent, but only get turned on in particular areas. So how can you conceptually think of doing that? And that I think requires bifunctional systems where maybe some external radiation, who knows what it might be, would mean that you can turn these things on in particular regions, but everywhere else they're off. And so the networks like the Nanomedicines Innovation Network really allow you to get that sort of expertise together to build the next level of sophistication of the delivery systems. So, from a Canadian point of view, this is something where we are at the moment leaders in this. and we need to. This is a way of, at least we're going to be in the game for some time with that kind of contribution from people with much different backgrounds.
Any other point?
No, one other thing is that this is, with the success of OnPatro, this is the result of literally hundreds of people's work over 20 years.
Yeah.
So you don't do much by yourself. It always has to be in concert with a whole lot of other people that are, doing various other things. And the collaboration we had with Alnylam from 2005 to 2012 was one of the best collaborations I've ever been part of. And we also had our local company, Acuitas, that was involved in that as well. But it's people, very, very motivated and dedicated people working towards this common aim that have managed to get us to the point where you actually have an approved drug in a new RNAi therapeutic. But it's it's essentially a real team effort. There's no way you can do this by yourself.
Yeah, that's nice words to the end.
Okay.
I hope everyone enjoyed this interview. You got advice and stimulation and motivation for the future. It's, I think, a really exciting future for non-viral gene therapy. And thank you for everyone who are part of the CRS focus group for gene delivery and genome editing. If you're not a member yet, go ahead and register. And thank you, Pieter. Thanks a lot for your time. And
well, thanks, Nam. It's been a pleasure talking with you.
Thank you.